Thank you for joining me in this advanced bioreactor module. If you watched the previous, more basic module, you'll see that now we're going deeper into the science, engineering, and design of wood chip bioreactors, starting with one of the most important concepts for denitrification, which is dissolved oxygen and oxidation reduction potential. In the introductory lesson of the Bioreactor Basics module, we learned about the 4 things necessary to have denitrification occur in a wood chip bioreactor. One of those was anoxic conditions, and so in this video, we'll review some water chemistry to explore what exactly that means. One of the most important water quality criteria in general when we talk about the status of water bodies is the amount of dissolved oxygen present. DO is important for pretty obvious reasons. I think, for example, aquatic creatures need dissolved oxygen in the water to survive, and a good rule of thumb is that for most fish, you want at least about 5 milligrams DO per liter. That's species dependent and growth stage dependent, but that's a good rule of thumb. Another rule of thumb is that hypoxic conditions, that is, low oxygen conditions, are considered to occur at DO concentrations of about 2 or less. And you see that illustrated here from the 2022 measurements of the Gulf of Mexico hypoxic zone. The DO levels of less than 2 measured at the bottom of the Gulf are in those red colors. I've been talking about dissolved oxygen as a concentration, but you should also know that it's often discussed as a percent of saturation. And if you have a DO probe, it will commonly read in both of those units, concentration and percent of saturation. If you want to convert from DO concentration to percent saturation, there is a nomogram to do this conversion. How cool is it to use a nomogram? I mean, there's also online calculators that can do this pretty quickly online, but let's take it old school. A nomogram is a diagram that represents the relationships between 3 or more variables by representing them on a series of scales. And you just use geometry to relate the variables across the scales. For example, say we measured the DO of water that was 22 degrees Celsius and it was 8.5 milligrams DO per liter. You just draw a line from 22 degrees to 8.5 and you see it goes right through right about 95% saturation. So you know that at that DO concentration and water temperature, you have 95% saturation of DO in that water. For reference, that 95% is pretty good in terms of relative water quality. Remember, our rule of thumb for fish was a concentration of 5 milligrams per liter DO, and so our 8.5 milligrams per liter in this example is pretty good relative to that rule of thumb, and so this jives in terms of percent saturation too. The last thing I'll mention about dissolved oxygen is that it is not the only gas that dissolves in water. In fact, many gases dissolve in water, and the quantity of any gas that dissolves into a solution is controlled by these 4 factors. I'll just go through a couple of these, starting with partial pressure of gas in the atmosphere. When you're at 1 atmosphere, like at most locations on the Earth's surface, the partial pressure of a gas equals the mole percent, or mass fraction, of that gas in our atmosphere. That's what I'm showing in this table. I make this point because, like I said, oxygen is not the only gas that dissolves into water, and later on in this module, we'll talk about nitrous oxide and methane, which are both greenhouse gases. Nitrous oxide and methane can be dissolved in the water that enters and leaves a bioreactor. The water temperature is really interesting because cooler water holds more dissolved gases. This seems somewhat backwards to me, but nevertheless, this graph illustrates that increasing temperatures, as you move from left to right, the solubility of oxygen in fresh water decreases. And you see that line regardless of the pressure, which are the 3 different lines. That means that cool water naturally can hold more dissolved oxygen compared to warmer water. Also, a side note, remember the nomogram from a minute ago? The percent saturation of dissolved oxygen in water, like we were trying to calculate, has to do with the solubility of oxygen in water, which is why we also have to have the water temperature included in this nomogram, because temperature impacts solubility of gas and thus also impacts percent saturation. This last bubble about impurities in the solution is also illustrated with this set of graphs. You see fresh water on the left and saline water or ocean water or marine water on the right. Comparing a given point, you see that at any given temperature and pressure, for example, 1 bar of pressure and 10 degrees Celsius, the saline water will hold less oxygen at saturation. The solubility of gases in water decreases as there's other dissolved materials present in that solution. When DO is present in water, like what we've been talking about, we can measure it and know that the water will support aerobic organisms. However, when the DO meter reads 0, we know conditions are anaerobic, but there are actually many different processes that can occur when the DO is 0. How do we know what processes are occurring when our DO meter only tells us 0? This is where the concept of oxidation reduction potential, or redox potential, comes in. I've said before that denitrification is an oxidation reduction reaction, or a redox reaction, where nitrate is reduced because it gets the electron and carbon is oxidized because it gives the electron. From a biochemistry perspective, the important thing about these reactions is that all redox reactions release energy. And that's why these reactions matter to the bacteria that do them, because the bacteria involved want to get the most energy that they can out of any process that they invest in. Different redox reactions release different amounts of energy. Redox potential sensors, or ORP, oxidation reduction potential sensors, are a way that we can measure a liquid's capacity to release or accept electrons. This means we can measure the potential for different redox reactions to happen in solution, like in water flowing through a bioreactor. ORP readings are in units of millivolts, electrical units, which makes sense because we're measuring the potential to transfer electrons, just like electricity. Here's an illustration of redox conditions in a wetland soil profile. As you get deeper and deeper in that wetland soil, in this example, you get increasingly reduced conditions. These ORP ranges get more and more negative. Remember, you can't go less than 0 DO, so this illustrates the usefulness of measuring and understanding ORP. For example, as you go down, you see the types of processes change from aerobic processes, where you still have DO in the water and the redox potential is the most positive, to nitrate reduction, which is denitrification, where our redox potential hovers right around 0 from negative to positive 50 millivolts, to sulfate reduction and then methane formation, which are both strongly anaerobic processes and have much more negative redox potentials. Showing these biochemical reactions a little more plainly by ORP, we often say that denitrification is a process that is done by facultative anaerobes, which means they can switch from breathing, or in other words, reducing oxygen when there's DO present, to breathing nitrate, or reducing nitrate, when there's no oxygen present. When there's no oxygen present and these bacteria switch to breathing nitrate, that is the process of denitrification. However, the denitrifying bacteria get more energy from the reaction when they breathe DO, that is, aerobic reactions. So they prefer to reduce oxygen rather than nitrate. When you look at this entire chart, the amount of energy released from each reaction decreases as you get more and more reduced conditions. When we measure a DO of around 0 and an ORP of between plus or minus 50 millivolts, we know the conditions are right for those very flexible facultative denitrifying bacteria to have switched to reducing nitrate. And an ORP of right around 0 for this process is a good reminder to us that these denitrifying bacteria are right at the cusp of aerobic and anaerobic conditions and that they're facultative. We often refer to denitrification as anoxic to differentiate it from the processes further down in the table, which are also anaerobic but are much more strongly reduced. To sum up, it's not just about having 0 DO in bioreactors. Yes, we need the DO to be very low, but it's more nuanced than that, and ORP helps us know what types of reducing environment we have. We need to be in the right range for denitrification. See you next time.